Hi everyone, welcome to this revision video on the structure of DNA and RNA. This is based on the A-level AQA biology specification, specifically point 3.1.5.1. Mind that I'm a bit sick, so you have to bear with me, but we will get through this together. So let's first look at some key definitions you need to know. DNA is an important information-carrying molecule which holds genetic information. A DNA molecule is a double helix with two polynucleotide chains held together by hydrogen bonds between specific complementary base pairs. RNA is an important information-carrying molecule which transfers genetic information from DNA to ribosomes. An RNA molecule is a relatively short polynucleotide chain, and a double helix is two strands held together by hydrogen bonds. Moving on to the terms. DNA is the abbreviation for deoxyribonucleic acid, and RNA is an abbreviation for ribonucleic acid. Polynucleotide is it's an equivalent to using the term polymer of nucleotides. And when we refer to complementary base pairing, we're talking about adenine with thymine or uracil, let me get my words out, and cytosine with guanine. And when we refer to a nitrogenous base, you can use equivalent terms such as organic base or nitrogen-containing base within your exam as well. Moving on, let's consider monomers and polymers when we're talking about these nucleic acids. So for the monomer of RNA and DNA, we're looking at nucleotides, which we will see the structure later on in these slides. And for polymer, we're looking at polynucleotides or nucleic acids, and these themselves are the DNA and the RNA. So nucleic acids, there are two types of nucleic acids, DNA or RNA. And now we're looking at the nucleotide structure, as I mentioned before. Here you can see the main structure of a nucleotide contains a phosphate group, a pentose sugar, and a nitrogen-containing base, which we mentioned. There are alternate terms for using, but here's the one that's on the graphic that is shown within the specification. Specifically, you will need to know this for your exam. The components slightly differ per different nucleic acid. So for DNA, its nucleotide contains the pentose sugar called deoxyribose. It still contains a phosphate group, and the organic bases it includes is adenine, thymine, cytosine, and guanine. And for an RNA nucleotide, for the pentose sugar, you now have a ribose, and there's still the phosphate group. And then for the organic bases, you have adenine, now uracil, cytosine, and guanine. Moving on, the characteristic bond for nucleic acids is a phosphodiester bond, unlike the other videos you've probably seen. And if not, make sure you watch all my other videos on biological molecules that have come before this and that will come after this. We don't actually have to draw out a phosphodiester bond, but just to know the name and what structures it goes between. So as it shows in this diagram, the phosphodiester bonds, which are labeled in red, form between the pentose sugar of one nucleotide and the phosphate group of another nucleotide. Moving on, let's talk about the structure of DNA. It is a polynucleotide or polymer of nucleotides, as we mentioned before. Each nucleotide is formed from deoxyribose, a phosphate group, and a nitrogen-containing base. Phosphodiester bonds occur between adjacent nucleotides. It has a double helix structure, and it also has hydrogen bonds / complementary base pairing between adenine, thymine, cytosine, and guanine. And here you can see a structure of DNA, and there's a few points that I really want to point out, so I'm going to get out my handy laser pointer just to make them very clear. If you can see, I'm just going to zoom in. You see these little notations here, which is a 3 with a little apostrophe and a 5 with a little apostrophe. All that means is, well, we refer to them as 3' and 5'. All that means is if you saw a diagram and you saw one of the strands ends with a sugar, and obviously it's a pentose sugar, so it's gonna look, if you're watching the video, this will be abundantly more clear. So if you are listening to the audio, make sure that you hop onto YouTube at one point to see the actual video of this. If it ends with an OH group or perhaps pentose sugar, then you would call that end the the 3' end, but if it ends with a phosphate group, as you can see as I'm circling right now, you would call that the 5' end. And other notable things is that adenine and thymine / uracil form 2 hydrogen bonds together. So the percentage of adenine within a molecule of DNA or RNA would be equivalent to the percentage of thymine or uracil within the molecule. And cytosine and guanine form 3 hydrogen bonds. So the percentage of cytosine would equal the percentage of guanine within these nucleic acids. Moving right on, let's look at the structure of RNA now. RNA is a polynucleotide. Each nucleotide formed from a ribose, a phosphate group, and a nitrogen-containing base. Phosphodiester bonds occur between adjacent nucleotides, and RNA is single-stranded. And as you can see on visuals again, so if you're on Spotify, hopefully you can see the video. If not, go to YouTube and you will see the structure of RNA, and you can see all the possible bases you can have. And once again, RNA contains the nitrogenous base uracil, whereas DNA contains the nitrogenous base of thymine. Moving on, let's do a quick comparison between DNA and RNA. And obviously there are quite a few differences between them, but these are the key ones that the exam board wants you to mention. So whereas DNA is considered very large or longer, RNA is considered very small and shorter. DNA is a double-stranded molecule / helical, whereas RNA is single-stranded. DNA contains thymine, whereas RNA contains uracil, and DNA contains deoxyribose, whereas RNA, oh my goodness, contains ribose. And moving on. And ribosomes are formed from RNA and proteins, and this will become increasingly more obvious as we progress throughout the course. And that is done with this quick revision video. I am trying to make them a bit faster because I know you are out there busy revising. You just want to get all that information through. I hope this video helped, and make sure to comment if you have any more suggestions of things you want to learn. I am trying to go through the course in chronological order, but obviously if there's key topics you want me to hit, I will obviously do those videos first. So thank you and happy revising.